Hello everyone and welcome to Chapter 10, Evolution and Natural Selection. Of course, we start with our learning objectives, and I again believe you can read through these if you'd like to. So how we're going to start talking about evolution is by putting forth a situation that we want to create. In this instance, we want to breed flies that are able to withstand going without food for more than 20 hours, because that is, as you can see from this little chart here, the average starvation resistance time period. So after about 20 hours, flies really die if they don't have any food. So how can we get flies to that point? Just to give you an idea of how long humans can go without food, the longest was a gentleman in 1981 named Bobby Sands, who was going on a hunger strike, and he made it 66 days just drinking water, and that is, his body wasted away and all that not so good stuff. But let's talk about how evolution comes into all of this. So what evolution is genetic change in a population. In this instance, as far as our experiment goes, evolution is being forced. Typically, when you think of evolution and how evolution functions, it's sort of a natural process for most species to be successful in their environments or to be successful in mating. Regardless of what evolution's purpose is for changing an organism, it is change within a population. Do keep in mind that individuals do not experience evolution, it's populations. So with our fruit flies, as far as inducing evolution, what we would do is we would take our population of fruit flies, remove the food, we'd come back at about 20 hours, see who's still alive, and then we would have them breed. We would only breed the most starvation resistant flies. In that case, if you kind of turn your attention to evolution in action and look at the results, for our first generation, or generation 0, the original population, they live about 20 hours. But if we start breeding the flies that live just past 20 hours, then generation 1, just one round after selection, those offspring will live 3 hours longer than their predecessors. And if we keep doing that over and over and over and extending that time period past that original 20 hours, and we keep breeding the individuals that last longer than that time, and we get to generation 60, the average starvation resistance time period has now jumped from 20 hours to 160. And that is a genetic change in a population. Again, this is being forced by humans in this experiment, but regardless, this is, in a way, how evolution works. So how does evolution occur? It's a consequence of certain individuals in a population being born with characteristics that make them more likely to survive, which is something we call natural selection. Now, natural selection, a lot of people sort of think of it as, survival of the fittest. That is not necessarily true. We're going to talk about that phrase here in a bit. But natural selection is more so like who's better at evading being somebody's lunch, who's best at surviving predation, who's best at getting the girls, if you will, who can reproduce more and reproduce the best. So if you can't do those things, then natural selection will weed you out. But if you are able to maintain these certain characteristics that allow for your genes, your genetics, to be passed on to further generations, then natural selection will work in your favor. So what types of changes can evolution cause in a population? And we're going to talk about the mechanisms of evolution here in just a little bit. And there are 5 primary lines of evidence for evolution and natural selection. And those 5 primary lines actually come from 5 guys. So some of these gentlemen you've probably heard of. I know one probably definitely for sure, but we're not going to talk about him until just the end. So we're going to start with the fancy fella over on the far left, Orges Louis Leclerc, Comte de Buffon. And if I said that wrong and you've taken French, then I am so sorry. Regardless, he suggested that the Earth was much older than previously believed. In fact, he estimated that the Earth was at least 75,000 years old, which is the minimum amount of time for the planet to cool from a molten state. And it was pretty well established based on rocks that the Earth had been molten hot, essentially. And that's what he said. Not a lot of people believed him, because well into the 1800s, the Bible was the sole doctrine explaining natural phenomena, and it estimated that the Earth was a very young 6,000 years old, and that the species and various things on Earth had not really been changed, which, of course, you know, we know from a lot of these gentlemen that the Bible isn't very well supported by the data and the hard facts that we can see in the Earth and from fossil records and various things. So speaking of fossils and things of that nature, that brings us to our next gentleman, Georges Cuvier. He stated, from finding fossils at the bottoms of coal mines and slate mines, that it showed that extinction had occurred, which is really would have been groundbreaking, notice the pun, groundbreaking evidence of basically that creationism was not necessarily what had happened, and this is the start of evolution starting to sort of form in some minds. The next gentleman is Jean-Baptiste Lamarck. He suggested that living species might change over time. He was kind of wrong in how he phrased things and how he thought it had to have worked, but what he ended up causing was a discussion amongst other scientists at the time. So moving on to Charles Lyell, he argued that geological forces had gradually shaped the Earth and continue to do so, and basically just that Earth was in constant changing states. So what his big thing was that the mountains and valleys and rivers and lakes and things were caused by, you know, tectonic plates moving and creating these landscapes. And that it was still happening, which, because of him, Charles Darwin actually started thinking up the idea of evolution. And I know you probably know who Charles Darwin is. We are going to talk about him quite a bit. So living species gradually change over time, and this, again, was a statement, an idea that started with Georges Cuvier, who discovered those fossils. And what he saw in those fossils is they were animals or plants that were unlike any living species that he could find or see. and especially in areas where there was nothing that looked like that. For example, these giant sloths or woolly mammoths, or elk that have antlers that are just almost double the size of their body. Essentially, he was just finding these things that looked probably like monsters to him at the time, but it made him start thinking about where these animals went and where they had to have gone, which is extinct. They all would have died out or changed into something else. All right, so moving on to a little bit more about Charles Darwin. So if you know a little bit about Darwin, he was born in England in 1809. His family was relatively wealthy, pretty well off. His father was pretty well educated and highly encouraged Darwin to go to college, essentially, to become an educated man like himself. So at age 16, Darwin went to the University of Edinburgh and was going to go into medical studies. As he was learning about medical practices and surgeries, he found that he just did not have an appetite for it. It made him very sick, and he really wasn't a fan, so he dropped out of that. From there, he went to study theology at the behest of his father at the University of Cambridge to become a minister. That also didn't really work out because his real love, instead of theology, was studying nature. He really loved looking at various species and organisms and all that stuff. He was really into it, that and like taxidermy. From there, he met the captain of the HMS Beagle in 1831, where he became the gentleman companion of the captain, which is to say sort of like a hired best friend is the best way I can kind of describe it. It's nothing romantic. It was quite literally just the captain felt that he was suicidal because his uncle had committed suicide, to which he just wanted someone to be there for him and kind of keep him on track. He also really, really liked Darwin. He favored him quite a bit, and it made a few other people jealous. Regardless, let's move on. So, while on the HMS Beagle and traveling around the world, Darwin ended up at the Galapagos Islands, which are a group of volcanic islands that have quite a few interesting species, such as giant tortoises, really, really docile lizards, which Darwin wrote that he had to step aside for lizards because they wouldn't move when he would go to put his foot down. He would constantly have to watch himself and not stomp on these poor lizards. And then the thing that really interested Darwin while he was in the Galapagos Islands was the birds, which that is kind of the big thing that he ended up studying. So, while Darwin was on the Galapagos Islands, he collected finches. His theory was that they were all the same species based on their physical characteristics, their traits. He made two very important observations: that the different island finches all resembled the mainland species, so they were all very, very similar. And that there was striking similarity between the fossils of extinct species and the living species in the same area, that he noticed a connection between fossils and living animals. While all of that is very good, he was wrong that these birds were the same species. They would have been similar, But upon further research from ornithologists, they weren't reproductively compatible, which means that one finch from one species cannot mate with another finch from another species, meaning they're two separate species. If they can't mate, then they are not the same species. So moving on to a little bit about how Darwin came up with the theory of evolution. He actually started thinking about it thanks to this book called the Essay on the Principle of Population by Thomas Malthus, which the book's main theme was that populations grow much faster than food supplies can match that demand, which Darwin was just reading the book for fun, just for entertainment purposes, which, you know, you'd kind of look at that and think that's not a very light read, but he was reading it for fun and it sparked some ideas. Darwin ended up saying that favorable variations would tend to be preserved and unfavorable ones would be destroyed, which is very much so a very loose definition of evolution. So in 1842, Darwin wrote a 35-page draft of his ideas, but he never published them. They remained unpublished for 14 years. He sort of just hid it in the back of his desk drawer. Charles Lyell, which we talked about before, urged him to publish first before anyone else did. But Darwin didn't really listen to him because he didn't want to be wrong. He also didn't want to be kind of the laughing stock of the community. So fast forward 14 years from the point that Darwin hid those papers in his desk and some sketches and all that good stuff. And we've got this young man, Alfred Russel Wallace, who independently identified the process of evolution by natural selection, which this is a really big thing that he sort of just figured out. Mind you, while he was delirious from malaria, he wrote Darwin a letter saying that he thinks that he's onto something, but he's a little nervous to put forward the information into the community. So that was in 1858. Darwin then says, "I got you, fam. Let's present the idea to the community together." And they did, and it was pretty okay accepted. I mean, the world wasn't necessarily ready for that change, so it wasn't graciously accepted by, you know, people who were very big on Christianity and the Bible, and it went against what was believed at the time. So Darwin then in 1859 published the works that he had written out 14 years prior to that, and beat Alfred Russel Wallace to the punch with publishing his book, The Origin of Species, which, if you think about it, kind of mean that he did that, but at the same time, he had figured it out loosely 14 years prior, and he just didn't want to be scooped with that by a younger biologist. So regardless, this was science-changing information that Darwin put out into the world, and it started a new line of thinking. That organisms change over time, some organisms have gone extinct and no longer exist, that Earth is older than 6,000 years, and that the geology of Earth is not constant, but always changing. So let's talk a little bit more about how evolution occurs. Now, what happens for evolution to occur is that the allele frequencies in a population have to change. If we look at this population of tigers, we've got orange tigers and we've got white tigers, which if you've ever seen white tigers, they're very pretty, but because they are overly inbred, in order to get more white tigers, you breed white tigers with white tigers to make white tigers. What happens is they're very unhealthy animals. They have a lot of health conditions. There is one tiger in particular named Kenny. He is a tiger that has Down syndrome. And if you look him up, he doesn't look like a healthy tiger the way that you would hope that they would be. So if we are looking at this pie chart, we're seeing a change in the proportion of pigment alleles in the population of tigers, which means that evolution is occurring. There is a change in that frequency. And when you have a change in the frequency of alleles in a population, that is evolution. And then just because we keep saying this term population, here's a little definition of it. So populations of organisms evolve, individuals do not, and populations are just groups of animals. This can be like a whole species. It could also just be referring to a particular group in a particular area. There are 4 mechanisms of evolution. So the first is mutation. This is an alteration of the base pair sequence in the DNA of an individual's gamete-producing cells that changes an allele's frequency. We've talked about how mutations are caused and how they arise through your DNA. We have this term called genetic drift. It is a random change in allele frequencies unrelated to any allele's influence on reproductive success. What that is saying is that you have a sudden event that occurs that means that a handful of alleles just aren't going to exist in the population anymore. Individuals have died, but it is of no consequence to how well they were surviving in their environment, how well they were able to reproduce. What this is like a natural disaster, like a forest fire or a tornado or a flood. This can also be a disease, like diseases and natural disasters are good examples of genetic drift. Taking out alleles, killing individuals in a population and taking those alleles out of the population. Next is migration. So this is a change in allele frequency caused by individuals moving into or out of the population. So this doesn't necessarily mean that the individual stays. With wolves, sometimes females will kind of go off on their own and stray away from the pack, where a lone male or a male from another I almost said pride, but it's definitely not a pride. Another pack may mate with that female, and then she will go back, have her pups in her original pack and population, and then it puts those new alleles from the mate that she had into the population. Or you can have individuals just up and leave and go to a different area, and that means that they're going to take their alleles with them. And then last but not least, we do have that wonderful term, natural selection. It is a change in allele frequencies that occurs when individuals with one version of a heritable trait have greater reproductive success, which is like the biggest thing that we want to get across here. In order to be successful in any capacity and for evolution to continue, you have to be reproductively successful. You have to be able to pass on that genetic information to the next generation, and then for them to take that and pass it on to the next, and so on and so forth. And natural selection is kind of encompassing sexual selection as well. It's a type of natural selection. It's whatever your heritable traits, so stuff that can be passed on. This is not always behavioral, usually it's physical. Whatever traits that you inherit that allow you to be better at reproducing, that's natural selection working in your favor. In the example that we have there, the little picture, there's a fox chasing rabbits. Rabbits, a heritable trait that they can get is their propensity for speed. So how well they can be faster or slower. Faster rabbits are going to be able to outrun predators. Slower rabbits are not. And by being slower, that means you're going to get eaten, which means you're dead, and you can't pass on that genetic information. So that would not be a good heritable trait because you'd die. So we are going to talk about a few of those mechanisms of evolution a little bit more closely. So a mutation here, a direct change in the DNA of an individual, is the ultimate source of all genetic variation. Incredibly, incredibly important. Because evolution is a change in the allele frequencies found in a population, mutations are going to alter the base pair sequences of an individual's DNA, and sometimes those mutations can be really good things, where they make it to where an individual is more reproductively successful, and therefore their alleles are going to be passed on, and those alleles will increase. So hence evolution and the way you want it to go. So mutations are random. You can't really force mutations to work in your favor, let alone the opposite. You can't force mutations to not work in your favor. And we can't predict ahead of time which individuals will have what mutations, and we cannot predict whether the consequences of a mutation will be benign, harmful, or useful. Sometimes they just don't do anything, meaning they're benign. So continuing on, talking about mutations. Mutation is the ultimate source of genetic variation in a population, like we said on that last slide. Very important thing to get across. It's a big one. Pretty much everything you see when it comes to evolution is stemming from mutations. So mutations are going to either are potentially not going to impact fitness, and we'll talk about what fitness is, or cause early death, which is not a good thing, or reduce an organism's reproductive success. Moving on to genetic drift. Remember, this is a chance event occurring and taking out alleles from our population. It can lead to fixation, which is an allele's frequency in a population reaching 100%. It just depends on what the event is that can potentially cause this event to occur. And there's a few different types of genetic drift. The first is the founder effect. This is genetic drift that occurs when the founding members of a new population have different allele frequencies than the original source population. So a good example of that in humans is something called polydactyl, or polydactylism, I suppose. This is a very rare condition where individuals will have more than 5 fingers or more than 5 toes on each hand or foot, respectively. It occurs at a much higher frequency among the Amish of Pennsylvania. And this is because the recessive allele causing it was carried by both a husband and wife in the founding population. Those founding members of the Amish in Pennsylvania were polydactyl. They had those alleles. So they then passed that on to their next generation, and that next generation passed it on until it became relatively commonplace amongst that community. So that is founder effect. The next effect is called population bottleneck effect. This is where you have famine, disease, or rapid environmental changes that causes the death of a large random proportion of the population. So this is what I was talking about, which is sort of what you should think of when you think of genetic drift, is those chance events that lead to death of individuals in a population, but it is of no consequence to the alleles and how successful those individuals are. It's indiscriminate killing, essentially. Okay, moving on to migration. So migration, also known as gene flow, is the movement of some individuals of a species from one population to another. And in this little example that we have two populations of the same species that exist in separate locations. And in this example, they are separated by a mountain range. And if some of those species from population 1 move over to population 2, they are then taking their alleles with them. So if they migrate across that mountain range, the individuals in population 2 will be increasing their alleles, while the population 1 will be decreasing their number of alleles. So when three simple conditions are satisfied, evolution by natural selection occurs. The three conditions are that there must be variation for a trait within a population. That variation must be heritable, so you must be able to get it from the parents. They must be able to pass it on to their offspring. Individuals with one version of the trait must produce more offspring than those with a different version of the trait, and that is called differential reproductive success. And we are going to look at each one of these conditions individually. So the first is variation for a trait. When you look at this picture, you probably see a lot of different looking people, but there's also a lot of different looking dogs. So variation is all around us all the time. And that doesn't mean that we don't share some traits. We certainly do, but there's a lot of traits that a lot of people don't share. And especially if you look at the population as a whole, there's a lot of variation in people and dogs alike. Variation is going to be that raw material on which evolution works. And this doesn't necessarily have to be something that is physically seen on the outside. It can also include biochemical variations. The second condition is heritability. So we call the transmission of traits from parents to their children through genetic information inheritance or heritability. Pretty self-explanatory. If you can get it from your parent, that is a heritable trait. Meaning like hair color, eye color, skin color, certain diseases, or certain affinities for various things, body types, etc., etc. And the third condition is differential reproductive success, and it breaks down into three parts here. That there are more organisms born than can survive. Organisms are continually struggling for existence, and some organisms are more likely than others to survive and reproduce. In our example, we have a little piglet who is the runt of the litter. He is the tiniest pig of his brothers and sisters, and he has a very reduced differential reproductive success because his more robust siblings are preventing him from accessing the mother's teats in order to get milk. So he's going to struggle substantially harder for food, which he needs to grow and thrive. And with his siblings being bigger and stronger than him, he's going to have a much harder time growing and thriving, and therefore has a reduced differential reproductive success. So this is just a little recap of natural selection. And what we've got going on here is just showing us the three conditions that need to be satisfied by natural selection if it is going to be occurring. So variation of a trait, heritability, and differential reproductive success. And this is just showing it as a summary here, so nothing crazy important you need to look at. So I did use this example earlier talking about natural selection. So here we've got slow, medium, and fast rabbits. Medium speed, that is, not necessarily like size. And this is going to be a trait that is varied across individuals, but it is a trait that is heritable. And what this trait is going to cause is differential reproductive success for each of the various parts of this trait. So the individuals that are slower are going to have less reproductive success because they're going to be killed off. But that doesn't mean all of them are going to be killed off. Some of them may make it through being the fox's lunch due to chance. There may be other factors in the way for that animal to have survived, but the likelihood of it surviving is not going to be very high. Its reproductive success is relatively low, or a chance for reproductive success, because it could be killed because it's slower. But the ones that are faster have a much higher reproductive success because they're going to actually live to be able to reproduce. That moves us into sexual selection, which again is sort of a subsect of natural selection. This is where some traits make individuals more attractive to the opposite sex. Typically, it's going to be females that make these decisions based on physical performance. It could be some type of mating dance, plumage on birds is a big thing. There could be various other instances. For humans, we're a lot more kind of picky, as well as it's not always the female's choice to reproduce with certain males. Males also make choices. That's not to say that males in other species don't, but humans are very kind of give and take. Both males and females make that decision, while most animals, it's typically just the female who is going to say yes or no to reproducing with a particular male. These traits are going to confer greater reproductive success. So the better these traits on animals are, say like peacock feathers, the bigger, brighter, more beautiful feathers on a male peacock will make a female peacock substantially more receptive to mating with him because he's got the goods, he's got the beautiful feathers. He will have a higher reproductive success for having that better trait than other males. The increase in frequency because they satisfy 3 conditions of natural selection, which we talked about just before. We are going to move into how evolution can be quantified, and that is through the Hardy-Weinberg Law. I'm going to give you a little bit of history with this before we get into what it all means. So R.C. Punnett asked G.H. Hardy, who was a cricket playing buddy of his, which, yes, that is the R.C. Punnett who made the Punnett square. He asked G.H. Hardy, words, guys, sometimes it happens. Do recessive traits gradually become less common in a population? To which Hardy was kind of like, yeah, I can answer this question. And the answer is no, that recessive traits do not gradually become less common in a population. They still kind of stay the same. And he thought he was pretty novel in making this statement. He was not. Wilhelm Weinberg, who was a much lesser known scientist at the time, had already published the answer to that question 6 months prior in a lesser known journal, to which G.H. Hardy became aware of because he had to be told that he wasn't the first one to think of this. So they got together and teamed up and answered the question with this law, the Hardy-Weinberg Law. So we have two factors here. We have P and Q. P is the frequency of the dominant allele. Remember, we displayed dominant alleles as being the capital letter. And Q is going to be the frequency of the recessive allele, which we do represent as a lowercase letter when we're looking at that. The way the equation works is that P plus Q will always and forever equal 1. That is the big thing that you need to get across and kind of understand here. Well, they went a step further, and they came up with the Hardy-Weinberg equation, or the Hardy-Weinberg equilibrium, as well as sometimes it's known. So this is a way that we can predict how common each genotype in the population will be, while P plus Q equals 1 is talking about the alleles, specifically the dominant versus the recessive allele. This particular equation has P squared plus 2PQ plus Q squared equals 1. Always and forever will equal 1, where P squared does not represent the dominant allele. It represents the frequency, so how common you're going to see the homozygous dominant genotype, so a capital letter and a capital letter. While Q squared is very similar, but it is how common you're going to see the homozygous recessive genotype, the lowercase letter and lowercase letter. And of course, we have to take into account our heterozygotes, which is 2PQ, which is to say that it's going to be our capital letter and our lowercase letter combined together. So here is an example of how the Hardy-Weinberg equilibrium can be used, and I'm going to try to go through this as best I can. This is a little bit harder to teach from behind a screen instead of using a board. So we are going to start here with the dominant allele, and what we want to figure out here is P. Now P is again going to be the allele frequency of the dominant allele. So what we want to set up first is going to be this fancy little F with our capital B there, and we want to know how much of this dominant allele we have. So first we need to know how many we do have. So how many capital B's are in our population first? So we are going to look at our population. In this instance, we are looking at some kangaroo rats, which they are adorable little animals. They quite literally look like a rat crossed with a kangaroo. We have our dark brown kangaroo rats, which have the homozygous dominant genotype. We have our light brown kangaroo rats, which have our homozygous recessive genotype. And then we have these special boys right here, which are going to be a combination of both the dark brown and the tan kangaroo rats, which are our heterozygotes. They have both a capital B and a lowercase b, a dominant and recessive allele. So from here, we need to figure out how many capital B's or dominant alleles we have in the whole population. How we get this is we take this number, 16, and we want to multiply it by 2. And the reason we want to do that is because each one of these individual kangaroo rats have 2 alleles. So they have 2 capital B's, 2 dominant alleles. So we want to take 16 and multiply that by 2, which is going to give us, 32. From there, we want to look at our heterozygotes, which these little heterozygote boys, they have one capital B, one lowercase b. They only have one dominant allele to give us for this equation. So we just take 222 as it is, and you can see that that's what's happening right here, is what they are pointing out there. So we have in total 32 plus 222, which is going to give us, let me do a little math here, 200 and, what is that? 34, I believe. Hopefully I'm right. So we've got 32, let me just double check. 32 plus 222. Okay, that looks like it works out. Nope. This should be a 5. My bad, guys. So we've got 254 capital B's in our whole population of 1,000 kangaroo rats. So what we are going to do is, to get this frequency here that we've written out, we want to take the sub part and divide it by the total number of alleles. So we have our subpart, which is 254. So we're going to take 254 and we're going to divide that by 2,000. Now, if you're wondering where we got the 2,000, it's because we have 1,000 kangaroo rats. That is our whole population. Each kangaroo rat has 2 alleles to give. So we're just going to take 2, multiply it by 1,000, and that's going to give us 2,000. So that's our total number of alleles. You can also take these numbers and add them up, and they'll equal 2,000. But you can also just make it easier and take the number of the population, how many you have, and then multiply that by 2, because they're all going to have 2 alleles, hypothetically. So if we take this number and we do the math, that's going to give us this lovely number right here, which is our p-value. Now, what you can do is you can do that whole process over and get the frequency for the recessive allele. So you just go through and, this sort of walks you through that process. Or, because we know that P plus Q equals 1, we can use this equation, because it will always be 1 in the end, that if we know P, we can figure out Q. So to figure out Q, we would take 1 and minus P, which is 0.127, which is going to give us 0.873. So again, you can get to that 2 ways, and you can always double check your answer. So hopefully all that made sense, and if it didn't, let me know. So moving on to this equation, so P squared plus 2PQ plus Q squared equals 1. This is how we would figure all of this goodness out. So they've kind of already done it for us. And the fact that we already know P and Q, we can actually figure this out pretty easily. First, we would square our P value, and that's what they're doing there. And then we would also square our Q value, which, is also right here. So we would square these values, and they would give us this value and this value. And then we'd want to figure out what 2PQ is. And you would just take the P value and the Q value and multiply them together, and then multiply them by 2. And that would give you this And that is how we get those genotype frequencies. And remember, when it comes to this equation, we're not talking about individual alleles, like dominant and recessive. We are looking specifically at genotypes. So remember that this is p squared. Heterozygotes are 2pq, and then our recessive boys are going to be q squared. There's a couple things that we need to assume when we are using the Hardy-Weinberg equilibrium, and that there is no natural selection occurring, no mutations occurring, no migration occurring, no genetic drift occurring, and that random mating is happening. So what random mating is that we're just eliminating sexual selection. So there's no female peacock choosing a male because his feathers are so gosh darn irresistible. It is completely random, and that is what we have to assume for these equations. So traits causing some individuals to have more offspring than others become more prevalent in the population. And this is where we have this term fitness. So it's a measure of the reproductive output of an individual with a particular phenotype relative to the reproductive output of individuals of the same species with alternative phenotypes. So this is everything to do with that term reproductive output. So how many offspring you are popping out into the world. And this is where I'm going to kind of talk about the term survival of the fittest. It's not necessarily that you are the best of the best of the best. You are the best at whatever gets you to be able to survive and reproduce. This could mean that you are the fastest or the smartest or the most timid because it helps you hide. It doesn't mean you're the bravest and the biggest and the strongest. That doesn't always mean that you are going to have a high fitness. If a mouse were to be brave, like have a very brave or very ornery trait, then he's probably going to die. So bravery is not going to be a way to be surviving and passing on your genetic information, especially if you're a mouse. So there are a couple elements important to an organism's fitness. So I believe this is the first one. An individual's fitness is measured relative to other genotypes or phenotypes in the population, where traits that confer the highest fitness will increase in frequency. The second is that fitness depends on the specific environment in which the organism lives. An organism's fitness varies across habitats. The example we have here are two mice, or two colors of mice, where in a lighter habitat, the allele for light-colored fur is fitter and increases in frequency, so its ability to pass on its genetic information to its offspring is going to be much higher in that lighter environment than the darker colored mouse. The darker mouse will then thrive like the lighter mouse did in the lighter environment, but the darker mouse will thrive in a darker colored environment, so where it is fitter and increases in its frequency. The third element is going to be that fitness depends on an organism's reproductive success compared with other organisms in the population, and reproductive success determines whether a trait's frequency will change. So again, that phrase, survival of the fittest, is quite misleading, and you may have used it, and in the way that you used it is probably correct, but when it comes to evolution, this term is not the most fitting. So some organisms in a population can become better matched to their environment through natural selection, and this is something we call adaptation. It's the process by which organisms become better matched to their environment, and the specific features that make an organism more fit will become more prevalent. So a lot of organisms adapt to their environments very well, and if you don't, you die. So it just sort of depends on that particular situation and that particular environment with that particular species. Natural selection does not lead to perfect organisms. The purpose of adaptation is not to get to an optimal endpoint. It's to be able to change with the environment and be able to change for food sources or whatever it may be. So in this example, we have the average beak size within a population continuing to fluctuate according to food supply. When in dry years, finches had to eat large hard seeds, and then bigger beaks were selected for. During the wet years, finches could eat small soft seeds, and then smaller beaks started becoming more of a thing. So it just depends on the environment and those animals' ability to adapt and become better at surviving in those environments. So some factors that prevent populations from progressing inevitably toward perfection. The first is that environments change very quickly, especially with humans. There's a lot of environments that change because of humans, like the rainforest and various other places where deforestation occurs. So just environments change, and animals can't evolve fast enough or adapt fast enough to become, perfectly adapted for an environment. Everything's changing. So will the animals. They will never reach that perfect pinnacle, peak, body, essentially. The second is that variation is needed as the raw material of selection, which we talked about before, and there may be multiple different alleles for a trait, each causing an individual to have the same fitness. So that's saying that there's not just one trait that's going to be fantastic for an organism to survive in a certain environment and have pretty good fitness. There may be multiple traits that allow for the same species to thrive and have the same exact fitness. So not everybody's going to be the same, but they could have the same fitness. Artificial selection is a special case of natural selection. How I think of this is forced evolution, or man-made evolution. So plants and animal breeders unknowingly understood natural selection long before Darwin. We've been doing it for a long time, regardless of if we knew what we were doing, like the mechanism of how we were doing it or putting a name to it. We've been breeding animals and plants to kind of serve our purposes, which is called artificial selection, that creates animals or plants with exaggerated versions of desired traits. Corn is a fantastic example of this. We've been breeding corn to produce better kernels, more edible kernels. You can think of seedless bananas. We've been breeding them for that purpose. Certain animals, we breed them for those purposes. Cows that produce milk, so dairy cows, we produce them, or they've been developed so that they produce milk longer, and also the quantity of milk is increased as well. So you breed animals and plants to get that end result that's going to be what you want, that's going to be there for consumers and consuming and all that good stuff. But that is still artificially selecting and creating like man-made evolution with these species. So natural selection can change the traits in a population in several ways. There are three that we are going to talk about: directional, stabilizing, and disruptive. The first is directional selection. This is where individuals with one extreme from the range of variation in the population have higher fitness. The example is turkeys. on poultry farms can no longer mate naturally because their breast muscles have become so large. Artificial insemination is required for the reproduction of turkeys. The next is stabilizing selection. This is where individuals with intermediate phenotypes are most fit. And then the last is disruptive selection, where individuals with intermediate phenotypes have the lowest fitness. So up to this point, we've only really seen evolution occurring on physical traits, but evolution can also affect complex traits and behaviors. In this instance, we have a natural selection occurring for slow learners versus fast learners. So after 6 generations of selection, the rat selected for fast learning made only half as many errors as the slow learners. So you can see on that green line that rats that were slow learners were making the most errors in the maze, are selected and bred with each other, and they got progressively more slow. As opposed to the fast learners, which made fewest errors in the maze, were selected to breed with each other, and they basically maxed out with making so few errors in the maze by the 6th generation that it basically plateaued with how many errors the rats would make. It's really kind of cool that you can select for these types of traits and behaviors, which would be something that would be fairly complex and not something that was necessarily physical. Just very cool. So if up till now you're not completely sold on the theory of evolution, there are 5 primary lines of evidence that support that evolution exists, and we're going to go through each one. So we are going to start with the fossil record and then work down our list. So the fossil record is one of the 5 primary lines of evidence demonstrating the occurrence of evolution. Fossils are the preserved remains of organisms from long ago. And we can see a pretty good lineage of animals based on the fossil record. Dinosaurs really stand out in that. We can see that they existed, and we can kind of see where they went, which is into quite a few birds. So we can see that progression, and also that, due to various extinctions, we can see how animals progressed and either continued to progress and change through time. We can see very similar traits on fossilized animals versus animals that exist today. We can also see where lineages stopped. So there's nothing like that animal anymore. That extinction process completely wiped out that lineage, and that also helps support evolution. The animal that you see is Archaeopteryx on the screen, and it shows a transitional form between modern birds and reptiles, which is really cool. We can see the feathers, which Archaeopteryx is one of the first animals that we saw feathers, because feathers don't fossilize very, very well. So up to that point, it was sort of pretty well known, and especially during the making of Jurassic Park, that those animals potentially would not have had feathers. We know now that there's a good chance there were quite a few dinosaurs, at least from the ones that we saw in Jurassic Park, that potentially had feathers. More so than what we thought prior to that. Still with the fossil record, we have something called radiometric dating, which is not a newfangled type of Tinder or Bumble or anything like that. Radioactive isotopes are going to break down into stable compounds at a constant rate, and dating is done by evaluating the amounts of a certain radioactive isotope presenting in a fossil containing rock. So we can see how old something is based on how many radioactive isotopes there are present. So it's kind of a nice way of measuring how old something is. Continuing with fossilization, it's really interesting to see that fossilizing something is not as common as you might think. It doesn't happen as much as you might think. Compared to bones that have been found of early hominids, so early humans, there's only like a handful, like in the single digits of fully fossilized Tyrannosaurus rexes. There's so few that it might really shock you to know that there really isn't it isn't that common of an event. But when it does happen, it represents that organisms that happen to live in a location were present at that time. We can see a lot of fossilization in certain areas that really support the idea that the continents were all one, like Pangea. So that really helps support that. Biological remains that could be preserved under certain chemical conditions, and only the physical structures that can leave fossils will leave fossils. Like I said on the last slide, feathers don't fossilize well. And so Archaeopteryx was like a huge event finding that, because it preserved those feathers. Again, that doesn't normally happen. And then moving on to well-preserved fossil records. One of the examples we have is of horses or equids. The evolutionary history of horses is among the most well-preserved in the fossil record. Due to this record, we know that since horses first appeared around 55 million years ago, there have been many branches to their evolutionary tree, a sample of which is shown here. And you can see from their ancestor, Hyracotherium, that it's taken a few individuals to get to the horse that we have today, and quite a few of these intermediate stages that we have of these equids are now extinct. They do not exist anymore because it got us to the horse that we have today. But it, of course, has taken 55 million years to go from that original horse, quote unquote. It probably wouldn't have necessarily reminded you of a horse if you had seen it. But now it's gotten to be the horses we have today. And then even from there, we've started breeding horses with various other close relatives and creating even new species. And horses themselves have been sort of, dividing amongst themselves. Like you've got, the really tiny little ponies versus, like the big Clydesdales and stuff. And there's a good chance that we're going to have even more speciation amongst horses at some point as time goes on. And the last little thing about the fossil record is structures like I don't want to say like a fully formed hand, but like the almost hand-like structure with wrist bones of lobed fin fish and something called tiktaalik versus early amphibians. So we can see through the fossil record this transition, but also very similar features of a fish to a very early amphibian, and then the intermediate stage there. This discovery was really cool. You've probably heard this term, the missing link. You know, everyone kind of referred to like chimpanzees, and like Tarzan was the missing link, that kind of thing. In reality, it's Tiktaalik. Tiktaalik is this adorable boy on screen. He is the intermediate stage of very early fish to amphibians that was able to start crawling intermediately out of the water and onto land. And we can see how closely related Tiktaalik's quote-unquote hand and wrist look to be to a lobefin fish and an early amphibian that would have been crawling around on land. So it's just really cool to see transitional phases like this in invertebrates, especially that span between when animals were starting to climb out of the water and when they were living in the water. So super cool. Super cool. Tiktaalik, look at him. Moving on from fossils into biogeography. So geographic patterns of species distributions reflect species evolutionary histories. So biogeography is the study of the distribution of patterns of organisms around the world. So the example that we've got up here is the Hawaiian honeycreepers. The Hawaiian honeycreepers of Hawaii have adapted to a wide range of habitats, yet still closely resemble a finch-like shared ancestor found 2,000 miles away. So this sort of helps support that despite the distance, and those animals couldn't have flown that distance just for fun, this supports this theory of evolution because the ancestors that were quite finch-like would have arrived in Hawaii 4 to 5 million years ago when Hawaii was substantially closer to North America, and then started to evolve on their own to create their own species, but they still resemble that original ancestor that is found in North America. Marsupials are another really good example of this, and if you know what a marsupial is, they are animals that have pouches. So the Australian marsupials versus the placental counterparts of various other places around the world, because if you don't know, Australia is a continent, a country, and an island. It is kind of its own thing apart from the rest of the world, and is known for having a wide range of marsupials. Looking at sugar gliders in Australia, a lot of people have these animals as pets, and sugar gliders are very unique looking, but gosh, if they don't look just like a flying squirrel. So they're very, very closely related to flying squirrels with a lot of their features that they have, especially those, you know, big wings and adorable faces. But they're so far away from each other that the theory is that they come from a common ancestor. We can also see this with a few other animals in Australia versus placental counterparts that live in various other places, like the numbat versus the giant anteater. They're very, very similar, and yet a different species. And it no longer exists. The Tasmanian wolf, they are extinct. which is quite sad, versus the gray wolf. There's a lot of features that are very similar between these animals, and yet they would have been thousands and thousands of miles apart compared to potentially where they would have started with their ancestry. Moving on to comparative anatomy in embryology, where it helps us to reveal common evolutionary origins. So there are an astounding amount of shared embryological stages that indicate common ancestry. So all of these animals on the screen have gill pouches and bony tails at one point in their embryonic stage, and that includes a shark, a turtle, a chicken, and then a human even. So at one point in human development, as you are, an embryo, this is a very early stage of, becoming a baby, you have these things that all of these other animals have too, which helps support that we all have a common ancestor, which is likely to be Tiktaalik or some type of early amphibian or very early fish. So we're all connected. We're all family. Continuing with that idea that we all share certain things at those various stages, we can also see a lot of comparative anatomy in vertebrates as far as, and we've already talked about this, the kind of hand to wrist structure. So the bones that make up our hand and wrist are all pretty similar to how a lot of other animals have that bone structure laid out. So if you go back a few slides and look at the lobed fin fish, that hand-wrist situation compared to their next ancestor, which is Tiktaalik, which to our next ancestor, the early amphibians, and then you look at all of that compared to a human hand and wrist situation, to a horse, to a bat, to a whale or a dolphin there. What that shows us is that we have all of this anatomical structuring that is incredibly similar and has almost a base layout that is almost identical. So the way that these bones form to create the extremities, so hands and feet, is just very, very similar to each other, despite how different a human is to a bat, or a horse is to a porpoise, or all of these things are to a lobed-finned fish, So it's just, it really helps support that idea of evolution, that we all have a common ancestor. And through this process of evolution, yes, we have all deviated away from that original common ancestor, but you can't get rid of the basis of where we came from and how that anatomy, although it has changed drastically, still is similar to each other. It's just super cool. Can you tell this is my bread and butter? I love this stuff so much. So still talking about comparative anatomy a little bit here. We're going to bring up something called vestigial traits. So these are organs or structures that come from ancestral states in your lineage. In the case of little vampire bats, they have molars, and that doesn't really make too much sense because they have no reason to grind food, because they live on almost exclusively, well, not just almost, it is exclusively a liquid diet. And the reason that they have molars is because their ancestors did. Just like humans have wisdom teeth and an appendix. There's a few other things, a tailbone. We don't use those structures anymore, but our ancestors did. So finishing up with comparative anatomy, we have something called convergent evolution, which is going to produce analogous structures. So starting with different materials, eventually it comes to perform the same function through convergent evolution. And the best example we've got of that is wings. Wings is a very cool convergent evolution that produces wings in various animals. Moving on to molecular biology is going to reveal that common genetic sequences link all life forms. So all living organisms share the same genetic code, or at least really similar information, if not most of their genetic information is the same. Related individuals share a greater proportion of their DNA than unrelated individuals, and similarity can be measured by comparing DNA sequences. So when it comes to molecular biology, as far as showing that we have genetic similarities, we can look at hemoglobin, which is the protein that makes up red blood cells. We can look at the time elapsed since a common ancestor was last shared versus the number of amino acids in the beta chain of hemoglobin that are the same or different. So the last time humans had a common ancestor with an animal like a lamprey is a really long time. A lot of years, so millions of years, 564 million years have elapsed since humans and lampreys shared the same ancestor, hypothetically. And between the two, there's little to no similarity in their amino acids within their beta chain, which make up hemoglobin. So there's only a few that overlap anymore, compared to, say, a rhesus monkey, where humans and monkeys would be more closely related and having a common ancestor about 23 million years ago. There's quite a few similar amino acids still lingering on that make up hemoglobin, which really does make sense. So moving on to the lab and field experiments that help support evolution, we can look at antibiotic-resistant bacteria, which reveal evolution in progress. One of the biggest concerns when it comes to bacteria is that it becomes antibiotic resistant, and in hospitals you do get that situation happening a lot with something called MRSA, or MRSA, and that is bacteria that becomes completely resistant to antibiotics. It's not a good thing. So this is that process of evolution occurring of antibiotic-resistant bacteria. So in the 1940s, when first used as medicine, penicillin was uniformly effective in killing the bacterium Staphylococcus aureus. So you've got the penicillin in the middle of this petri dish. You probably are using some type of agar, which is going to help the bacteria grow, and you have this range or this ring around the penicillin where the Staphylococcus isn't growing, because that's the kill zone. The Staphylococcus is that white stuff that's around the edge. Today, the process of natural selection has led to an increase in antibiotic-resistant alleles within the bacteria, and now humans are relatively susceptible of getting infections by Staphylococcus. You've probably heard of a staph infection due to this bacteria that has become penicillin resistant or antibiotic resistant. So that's not a good thing. That is something to be concerned about for sure. And it's snuck up on us, but here it is. We have reached the end of Chapter 10. I hope you enjoyed this lecture. It's a little bit of a longer one, so I'm going to keep my goodbye short. As always, you can message me if you have any questions, but of course, make good choices.